This is my model of a cross-section of the cochlear duct, and this is representing the basal turn. So I'll first go through the anatomy, starting with number 1, we have the otic capsule. 2, we have the spiral ligament. 3, stria vascularis. 4, the scala vestibuli with the white beads representing perilymph. 5, scala media with the teal beads representing endolymph. And 6, the scala tympani with the white beads representing perilymph. 7, which is kind of hard to see, but 7 is down here, it's the osseous spiral lamina. 8 are the auditory nerve fibers, 9 is the spiral ganglion, and then 10 leading out to here would be the modiolus. 11 is Reisner's membrane, 12 is the tectorial membrane, and then this black line is the reticular lamina, and that's number 13. 14, this base below here is the basilar membrane. 14A is the arcuate zone of the basilar membrane, and 14B is the pectinate zone of the basilar membrane. And then finally, since this is the basal turn, 15 is the oval window, and 16 is the round window. Now for the anatomy of the organ of Corti, I do have it color-coded over here too. Orange are the inner border cells, the blue is the inner hair cell, yellow inner phalangeal cell, green are the rods of Corti, or those inner and outer pillar cells. The tunnel of Corti and the space of Newell are covered with these blue beads representing the cordolymph. The light pink are the outer hair cells, and then you can see that they're supported by the outer phalangeal cells or Dieter cells, which is the red. The purple are the cells of Henson. The reddish pinkish here is the cells of Claudius, and then the gray are the Böttcher cells. So that is the anatomy. Now I'm going to show you the materials that I used to create this. So the white otic capsule and spiral ligament was made out of modeling clay. All my fluids are represented by different colored beads. The auditory nerve fibers are made with strings. Some sections of the organ of Corti and the spiral ganglion are made of paper. The tectorial membrane is leather. The stereocilia, it's actually the head of a toothbrush, and I cut out the inside. And then the stereocilia of those inner hair cells are not touching the leather, which it's really hard to see that on the video, but in person you can clearly tell that the inner hair cell stereocilia are not touching, but the outer hair cell stereocilia are embedded in that leather. Also, the stria vascularis was made out of Play-Doh, and then the oval and round window are made out of balloons. And then I just used a string and some paper mechanisms to help move the structure. So now I'll show you how this structure actually moves. So it's kind of hard to do trying to hold my camera here, but when you pull the string, it moves the basilar membrane. And you can see the movement of those beads. And again, it's really hard to see those stereocilia, but the stereocilia of the outer hair cells are moving with the tectorial membrane, and those inner hair cell stereocilia are not touching. An area of this design that my group helped me with was the auditory nerve fibers. I wasn't exactly sure how I wanted to represent them. I was thinking it might be cool to do a little string of battery-operated lights, but my group reminded me that it's good to have a lightweight structure, so I just went with strings. And then when building the cochlea, I did have a lot of times when I had to stop and refigure things, and it was kind of difficult to figure out how to make it move, and there were a lot of trial and errors that occurred in the process of making this, but I'm happy with how it turned out. And I will say that I feel like I have a better grasp of the anatomy of the cochlea, so it definitely helped. I hope you enjoyed this video showing my cross-section of the cochlear duct.